This is the second set of PowerPoints under immunology. So we have the cellular components. Some of the cells here are just a review of the cells taken in the other topics. All cells of the immune system originate from a hematopoietic stem cell in the bone marrow, giving rise to two divisions or two major lineages. We have the myeloid progenitor cell and a lymphoid progenitor cells, and these are the precursor cells. These cells make up the cellular components of both the innate or nonspecific and adaptive or specific immune system. The next two slides will give us the different cells of the immune system from the myeloid lineup and the lymphoid lineup. So in this particular slide, we have the cells from the myeloid lineup. So this slide also will give us the two divisions of the cellular system of the immune system. Going upward, we have the myeloid line up, and towards the bottom, we have the lymphoid progenitor cells, which give rise to four types of cells. We have the lymphocytes, the T lymphocytes, we have the B lymphocytes, the dendritic cell, and the natural killer cell. Again, in the myeloid lineup, we have the myeloid cells. Particularly, we have the monocytes, the macrophages, the leukocytes, the megakaryocytes, and erythrocytes. In the lymphoid lineup, we have the lymphoid cells. So we have the lymphocytes, both the B cells and the T cells, the natural killer cells or NK cells, and we have the dendritic cells. Again, if we follow the arrows, we have the different cellular components of the immune system from the myeloid lineup and the lymphoid lineup. The myeloid cells are composed of reticuloendothelial system or the RES, under which we have the macrophage systems or the phagocytes. Then we have the monocytes and connective tissue cells involved in phagocytosis, and all of these are referred to as macrophages. The reticuloendothelial system is composed of both the free macrophages and fixed macrophages or histiocytes. The free macrophages are found in the mucous membrane of the digestive and respiratory tracts. They could also be found lining the blood sinuses of the bone marrow and also in the blood vascular system as part of the blood vascular cells, while the fixed macrophages or histiocytes are found in a particular organ or structure of the body, and they are considered to be the guardian of that particular organ or structure as a macrophage or a phagocyte. The blood vascular cells are both the red blood cells and the white blood cells plus the platelets. All of the cells described will be stained using the right stain, the most commonly used stains in differential smears. So the blood that are described in the following slides will be colored based on how they appear using the right stain. For the leukocytes or the white blood cells, all of these cells are involved in body defense mechanisms, and they are grouped into granulocyte or granulocytic series and agranulocytes or agranulocytic series. So we have here in the next slide the groupings of the white blood cells, and these are the granulocytes. We have the neutrophils, eosinophil, and basophil. Under the granulocytic series, we have the polymorphonuclear cells or PMNs, or simply neutrophils. So using the right stain, the granules of neutrophils are very fine azurophilic granules distributed throughout the cytoplasm. These granules are seen using a sophisticated type of microscope, not the ordinary compound microscope in the lab. The granules have affinity for the neutral stains. That's why they are referred to as neutrophils. And using a differential count smear, they are around 55 to 65%. The neutrophils are involved during bacterial infection. So they ingest and kill bacteria. So in the cells are lysosomes that releases a particular enzyme that digests bacteria. The next cell is the eosinophil. So they have affinity for the acidic stain or the eosin stain used in the staining of the blood smear. So eosinophil, affinity for the eosin stain or acidic stain. And because of this, they will appear as orange-red granules, and they are uniform in size that may cover the entire cell, or it will cover the nucleus of the cell. These orange-red granules are distinct morphological characteristic of the cell that will help us easily identify them under the microscope. Now, in a differential smear, they are just around 1 to 4%. The next cell is the basophil. Using the right stain, this cell have affinity for the basic stain. That's why they are referred to as basophil. The granules appearing in the cells are coarse blue-black granules of no particular size. They may be large granules or smaller granules. So they appear as coarse blue-black granules of varied sizes. Now, these granules may fill up the entire cell or may cover the cytoplasm of the cell. In a differential smear, usually they will be found 0 to 1%. For the agranulocytes, we have the lymphocytes and monocytes. So A stands for without granules. The lymphocytes are cells that will stain blue in color. The characteristic color of the cytoplasm of the lymphocyte using the right stain is robin blue in color. The nucleus is round and may fill up the entire cytoplasm, and usually the lymphocytes are involved during viral infection. In a differential smear, they're around 20 to 45%. Under the microscope, we cannot differentiate the different lymphocytes in the smear, so they could appear as small lymphocytes 4 to 7 micrometer or medium-sized lymphocytes 7 to 11 micrometer and large lymphocytes 11 to 15 micrometer. So they will appear morphologically the same. The monocytes are the largest of the agranulocytic series. So they differ in the appearance of the nucleus. Unlike the other cells of the white blood cell series, the nucleus of all the cells are condensed or they are compact, while in monocytes, the chromatin material in the nucleus are loosely arranged, and it will give us a staining characteristic of the nucleus as pale nucleus. Now, using the right stain, the cytoplasm will appear grayish in color, and sometimes in the cytoplasm, vacuoles are seen, indicating active phagocytosis. In a blood smear or differential smear, they are around 3 to 5%. As a summary, we have the granulocytic cells and the agranulocytic cells. So again, for the granulocytic cells, we have the basophil, we have the eosinophil, and the neutrophils. You can see that in the cytoplasm or in the cells are granules, while in the agranulocytic group, we have two cells, the monocytes and the lymphocytes without the granules. This is again the summary of all the innate immune cells of the body, or these cells are found naturally within the body. Other cells, together with the blood cells that were presented, are the connective tissue cells. So we have the mast cells and we have the histiocytes. For the histiocytes, we will just go through the different slides presented because this is just a review. We are through with the histiocytes in the other topic. The mast cells are concentrated within the respiratory and gastrointestinal tracts and within the deep layers of the skin. It releases a chemical substance, histamine, upon encountering certain foreign substances or certain antigens, thereby triggering an allergy or an allergic reaction or hypersensitivity reaction. The mast cells are resident cells of several types of tissues, and they contain coarse blue-black granules just like the basophils, rich in histamine and heparin. Their best role is in allergy and anaphylaxis, but they also play an important protective role intimately involved in wound healing and defense against pathogens. We have here the illustration of a mast cell wherein inside the cytoplasm are coarse blue-black granules containing the chemical substances. And also below, the interaction between the mast cells and other cells of the body. We have here an illustration of an antibody. We have IgE involved during allergy or during hypersensitivity reaction. So during this condition, the mast cell releases histamine and it causes the immunoglobulin E to interact with the granules in the mast cells, causing allergic response. We have here the interrelationship between a plasma cell and mast cell. So the plasma cell produces antibodies, and these antibodies are attached to the mast cell. Now, the mast cell with the histamine granules will release the chemical substance and it will cause an allergic or hypersensitivity reaction. For the histiocytes, this is just a review. So in the next several slides, I will just give few seconds for you to view the histiocytes as a review. The first histiocyte are found in the liver. We have the Kupffer cells. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Followed by the mesangial cells in the kidneys. Then we have the alveolar macrophages or dust cells found in the lungs. Then the LANGERHANS cells found in our skin. We have the microglial cells in the nervous tissue. It could be found both in the brain and spinal cord throughout the central nervous system. Then we have the littoral cells in the spleen involved especially during tumor of the spleen. The joints of the body also contains histiocytes, so they are found in the synovial membrane of the body. So focus your attention to the right side, upper right side of the slide, wherein we have the articular cartilage on both sides, and at the center is the synovial membrane. On the surface of the synovial membrane are cells we call the A cells, and these A cells have phagocytic function. An illustration of the A cells in the surface of synovial membrane. We now go into the lymphoid cells. From the red bone marrow in bones, we have the precursor cell or the stem cell giving rise to pre-B cells, which will develop into a B cell or B lymphocytes. together with the pre-T cells that will produce the T lymphocytes. So take note, the lymphocytes are produced in the red bone marrow, and the T cells, after they are produced in the bone marrow, will go into the circulation to the thymus gland for maturation and development, and again to the circulation as T lymphocytes, so they will go into the lymph node. While the B cells, once they are produced in the red bone marrow, will go directly into the circulation to the lymph node. So the lymphoid cells are the following: dendritic cells, B cells or B lymphocytes, T cells or T lymphocytes, and the natural killer or NK cells. We have here illustrations of the dendritic cells. So the dendritic cells have similar morphology as that of the undifferentiated mesenchymal cells, with their cytoplasmic extensions. So if they are present, we refer to them as interdigitating reticular cells. So they are involved in the different types of pathogens that will enter into the body. The dendritic cells were first described by Dr. Ralph Steinman. These are responsible for identifying pathogens such as viruses, fungi, bacteria, malignant cells, and they also function to present the cells or these foreign substances with their markers to specific T lymphocytes that will trigger the T lymphocytes to multiply and attack the pathogen. The dendritic cells are also capable of identifying normal dying cells and to present their antigens to different T cells that will tell the immune system of the body not to attack its own tissue. The, the dendritic cells were first described by Dr. Ralph Steinman. These are responsible for identifying pathogens such as viruses, fungi, bacteria, malignant cells, and they also function to present the cells or these foreign substances with their markers to specific T lymphocytes that will trigger the T lymphocytes to multiply and attack the pathogen. The dendritic cells are also capable of identifying normal dying cells and to present their antigens to different T cells that will tell the immune system of the body not to attack its own tissue. As mentioned, the dendritic cells are potent antigen-presenting cells or APCs that possess the ability to stimulate naive T cells to produce many T cells. They comprise a system of leukocytes widely distributed in all tissues, especially in those that provide an environmental interface. Maturity of dendritic cells will act upon phagocytosis of apoptotic cells providing either an immune activation or immunosuppression. The immature dendritic cells induces tolerance, while the mature dendritic cells initiate immune activation. These dendritic cells are mostly found in the skin and mucosal epithelium. We have the Langerhans cells of the skin. Unlike macrophages, dendritic cells can also recognize viral particles as non-cells, and they are also considered to be an APC, the antigen-presenting cell. Then from dendritic cells, we have the B cells. The B cells are also known as B cell lymphocytes. B cells spend their entire early life in the bone marrow, but upon maturity, their job is to travel throughout the blood and lymph circulation looking for antigens with which they can interlock. Upon activation, the B cells will transform to either plasma cells producing antibodies, or they will remain as B cells as memory cells. The B cells will develop once they will identify an antigen, and it will cause to replicate itself. These cloned cells will mature into an antibody manufacturing cell we refer to as plasma cells. The plasma cells are specialized B cells which churn out antibodies or produces many antibodies, more than 2,000 per second. Most of these cells die after 4 to 5 days, and those that will survive will become memory cells or memory B cells. We have here an illustration of the plasma cell, which its characteristic location of the nucleus, which is found at the peripheral part of the cell or at one side of the cell, with its cartwheel appearance of the chromatin material within the nucleus. We have here an illustration of the B cell proliferation or the replication of the B cells upon stimulation, and later on they will be converted to plasma cell to produce antibodies. So take note, it is the plasma cell that produces antibodies. The memory cells, as mentioned, are specialized B cells which grant the body the ability to manufacture more of a particular antibody as needed, in case a particular antigen is ever encountered again, or upon re-exposure to the same antigen. The T cell or the T lymphocytes are classified based on their function. We have the killer T cells, the helper T cells, the suppressor T cells, and memory T cells. As mentioned, the T cells are also known as T lymphocytes. Unlike the B cells or B lymphocytes, these cells leave the bone marrow at an early age and travel to the thymus, where they mature. And they can also recognize self and non-self foreign substances or antigen. We have here a list of the types of T cells or T lymphocytes with their characteristic and their function. So number one, we have the helper T cells. They assist in activation and transformation of B lymphocytes. Second, we have the suppressor T cells. They inhibit B cell activation and regulate immunoglobulin synthesis. The third, we have the cytotoxic T cells or killer T cells. These are involved on tumor cells, transplanted cells, and virus infected cells. So destroy all these cells. Then we have for number 4, the null cells or the null T cells. They lack surface immunoglobulin and they are also the non-marking cells. They participate in cell destruction with the aid of immunoglobulin. We have here in this illustration a cell that stimulate production of T cells or clones of T cells. So we have here the virus infected cells which will stimulate the T cell to produce more T cells. So we have several T cells that are formed. We have the killer T cells, the helper T cells, the memory T cells, and the suppressor T cells. We have here the roles of helper T cells. So it could either activate inducer T cells that will initiate differentiation of new T cells, or it can activate suppressor T cells that will shut down cell-mediated and humoral immune responses. The helper T cells could produce interleukin-2 that cause the production of cytotoxic T cells and cause cell-mediated immune response. It could produce cytokines and gamma interferon that will stimulate macrophages to congregate at site of infection, or it can bind to B cell antigen complexes that will stimulate the production of B cells and cause humoral immune response. As a summary, we have the lymphocytes in the blood circulation produced in the bone marrow, and we have two types. We have the B cells and we have the T cells. The B cells or the B lymphocytes mature in the bone marrow, then move to lymph nodes and recognizes antigens to produce antibodies or to remain as memory cells. While the T cells or T lymphocytes mature in the thymus gland and can be differentiated into helper T cells, killer T cells, suppressor T cells, and memory T cells. All of these T cells recognizes antigens. The helper T cells can stimulate B cell production and can stimulate killer T cell productions, while the killer T cells can cause abnormal cells to die or to burst. The suppressor T cells can turn off immune responses, and memory T cells can stimulate again production of B cells and stimulate the production of killer T cells. I think this is the last slide. And we are now going to the last set of PowerPoints, which is Immunology Proper 2.